So we'll now move on to the fourth speaker of the webinar, that being Dr. Amy Engevik. Amy is an assistant professor at the Medical University of South Carolina. Among her research directions, Amy's group is currently developing novel mouse models lacking functional myosin 5B to investigate how myosin 5B regulates bile acid secretion and bile acid signaling in the intestine and liver. We are very pleased to have Dr. Engevik speak today on this work in a talk entitled "A Targeted Metabolomics Approach for Quantifying a Broad Constellation of Bile Acids in Biomedical Research." The floor is yours, Amy.
Great. All right, can you see my screen?
We can. Thank you very much.
Right. Well, thank you for that nice introduction, and thank you for the invitation to present our work today. Like Andrew said, I am a mouse researcher, so I'm going to switch gears to more of using mass spec as a mechanism to understand what's happening in a mouse model of a human disorder. All right. So my lab studies myosins, and when most people think of myosins, they think of conventional myosins, which represent class 2 myosins. And class 2 myosins are involved in muscle contraction and are among the most widely studied myosins. However, there's a number of other myosin classes which represent unconventional myosins. And these unconventional myosins are involved in a diverse biological functions, which include organelle transport, cell adhesion, endocytosis, and exocytosis. And then movement of RNA, as well as signal transduction and cell motility. And so these actin motors, treadmill and F-actin, and really are helping organize the cellular structure, especially in a number of organ systems. So myosins are ubiquitously expressed throughout the human body, and there are about 39 myosin genes in humans. And our lab studies an unconventional class 5 myosin, known as myosin 5B, which is highly expressed in the inner ear, the reproductive tract, and the skin, as well as the intestine and the liver. And we're really interested in myosin 5B because inactivating mutations in myosin 5B causes a congenital diarrhea disorder called Microvillus Inclusion Disease, or MVID. So MVID gets its names because inside of enterocytes, which are the absorptive cells in the intestine, there are these microvilli that are normally on the apical membrane of cells. But when you have inactivation of myosin 5B, you instead have this internalization of these microvilli into what is called inclusions, which are basically rings. And this is abnormal. So patients with MVID really present with severe unremitting diarrhea that is universally fatal if there is no medical intervention. So all individuals with MVID end up having severe diarrhea, and this prevents the absorption of nutrients from food, which really hinders the amount of water that can be absorbed for dehydration for these patients. And this really impacts their developmental stages. They're unable to grow at a normal rate. They're incredibly unhealthy, and they all require total parental nutrition, which is IV nutrition. And this has a really high risk of sepsis. And eventually these children will need a full intestinal transplantation when they're healthy enough to receive one. And so even though the MVID is a rare disorder, it is incredibly has a very low survival rate, hovering around 25 to 50%. And in addition to all these GI issues, MVID patients have problems with their fragile bones, they have kidney issues, and there's a significant number of patients that present with liver cholestasis, which can progress to irreversible liver damage known as cirrhosis. And for patients with MVID, the current treatment regimen is total parental nutrition for years until they can have full intestinal transplantation. And now they are simultaneously doing liver transplantations to prevent the cholestasis from becoming irreversible and to stem off that problem. So it was originally thought that cholestasis in MVID patients was the result of prolonged use of total parental nutrition. However, recent studies have found that in humans, there's a unique mutation in myosin 5B that alone contributes to cholestasis without any GI complications. And there's been work in cell lines that suggests that myosin 5B is really important for hepatocyte function. So while the pathogenesis of MVID-associated diarrhea has become more clear, we still don't know very much, and there's no consensus on the origins of cholestasis in MVID patients. So we previously developed a mouse model with a germline deletion of myosin 5B, which I refer to as a myo5B knockout mouse. And these mice have many of the hallmarks of human MVID, including an inability to absorb water, resulting in early demise. So all of these mice die approximately 5 days after birth. And we also demonstrated these mice have significant intestinal abnormalities, which include fused villi, hyperproliferation of stem cells, as well as stented growth. And importantly, our myo5B knockout mice have the very characteristic inclusions, which you can see in TEM, and you can also clearly see in immunofluorescent staining where microvilli are marked in red, and you can see these rings inside of these enterocytes, which is what prior to RNA sequencing was used to diagnose all children based on pathology with MVID. So we know that in the intestine, myosin 5B is treadmilling on F-actin to deliver cargo bound to its tail domain to and from the apical membrane of enterocytes. And this is really responsible for delivering key transporters for water absorption. And when you lose myosin 5B, instead of having these transporters normally localized to the apical membrane, we have subapical accumulation and internalization, which results in decreased function and an inability for water to come into enterocytes and then into circulation for hydration. So we and others have shown how myo5B functions in the intestine. However, to date, no mechanistic studies or animal studies have connected changes in the gut with changes in the liver and analyzed the crosstalk between these two key GI organs in the setting of myo5B mutations. So my lab is interested in how myo5B functions in both the intestine and liver, with an emphasis on understanding the pathogenesis of myo5B-associated cholestasis. And again, to date, there has been no functional studies in animal models that lack functional myosin 5B looking at bile acid synthesis in the liver specifically. So initially, we started by determining whether myo5B is normally expressed in the liver and in which cell types they're expressed. And we queried the Tabula Muris single cell database for myo5B expressing cells. And we observed that myo5B is present in hepatocytes, which are the predominant cell type in the liver and are responsible for bile acid production and secretion. And when we look at expression levels, we can see that hepatocytes strongly express myo5B compared to other cells present in the liver, including endothelial cells, as well as immune cells like macrophages, natural killer cells, and B cells. Staining for myosin 5B, so we have hepatocytes that have a unique structure in the liver. So they're multipolarized, but they all surround these bile canaliculi, where bile is secreted from the hepatocytes into the bile canaliculi, where it will then go down the bile tree into where it's stored in the gallbladder. And so we stained for myosin 5B and MRP2, and MRP2 really marks the bile canaliculi. So the red, you can see these branching tree-like structures, which is the bile canaliculi, the apical membrane of hepatocytes, and myosin 5B is closely concentrated below that, suggesting that it might be important in the structure and function of the bile canaliculi apical membrane. So based on this, we hypothesized that myosin 5B may be responsible for the delivery of important bile acid transporters to the apical membrane or the bile canaliculi and hepatocytes. And thus, in the absence of functional myo5B, key bile transporters would be mislocalized, resulting in decreased bile acid secretion and contributing to the development of cholestasis. So looking at the H&Es of livers of control mice, we see the central vein in which hepatocytes are organized around, and they look well organized and healthy. But in our myo5B knockout mice, which are littermate controls for these or littermates for the control mice, we see macrovesical cytosis, where we see large lipid droplets in the hepatocytes. And we also see microvesicular hepatocytosis, which are smaller lipid droplets and represent problems with hepatocytes. We stained for the bile salt export pump, which is the major bile acid transporter in hepatocytes in control and myo5B knockout animals. And we find that in the BSEP that we have improper organization of these BSEP, and we have less branching and less BSEP positive cells compared to our control animals, suggesting that myosin 5B may be responsible for properly localizing BSEP and for the structure of the bile canaliculi. We looked at another important transporter that is present in bile canaliculi known as MDR1, which is the multi-drug resistance protein. And it's a xenobiotic transporter that's really important in a number of things, including drug absorption and in cancer therapy. So in the absence of myosin 5B, we see that MDR1 is clearly not properly localized and that we lose a lot of that biochemical structure, suggesting that myosin 5B might be transporting a couple of the key transporters that are involved in helping to concentrate bile and to deliver it outside of the hepatocytes. So we next compared RNA sequencing results from our mice to online databases of alcohol hepatitis, where we found that many of the genes upregulated during liver disease, especially severe alcoholic hepatitis, are also increased in our myo5B knockout mice compared to control mice. This might indicate that myo5B plays a key role in the maintenance of hepatocyte function and homeostasis, since it indicates there might be ongoing damage to hepatocytes occurring. So we next wanted to determine whether loss of myo5B really impacts the composition and functional capacity to produce bile acids. So there are two main bile acid synthesis pathways involved in conversion of cholesterol to bile acids in the liver. There's the classic pathway, which is initiated by CYP7A1, and the alternative pathway, which is initiated by CYP27A1. And in rodents, the primary bile acids formed are cholic acid and uracolic acid, and both the classic and alternative pathways equally contribute to bile acid formation in mice, while in humans, the classical pathway really predominates. So we looked at RNA sequencing from the liver of our control and myo5B knockout mice, and we see that we have altered expression of enzymes that are involved in bile acid synthesis. And importantly, we found decreased expression of CYP7A1 in myo5B knockout mice, which can downregulate bile acid synthesis. We also see that CYP7A1 is decreased, indicating that enzymes important in the classic pathway of bile acid synthesis are impaired by loss of myo5B in the liver. And increased CYP27A1 and CYP7B1 suggest that there may be compensation through the alternative pathway. But we really needed to look at bile acid composition to really delineate this. So after synthesis of bile acids, they are secreted from the hepatocytes into the bile canaliculi, where they mix with phospholipids and cholesterol to form bile, and they're stored in the gallbladder until bile is then released from the gallbladder in the presence of food into the intestine. And bile acids play an essential role in nutritional detergents, and they dissolve lipids and lipid-soluble vitamins. And primary bile acids are conjugated in the liver to the amino acids glycine in humans and taurine in mice. But when they enter the intestine, they are the conjugated bile acids can be deconjugated by bacteria, and then they form secondary bile acids. So we wanted to look and see what is the composition of bile acids in our control and myo5B knockout mice in the intestine and the liver. So we took ileal tissue and liver tissue from littermate control and myo5B knockout mice and homogenized them using 1.4 millimeters of beads of methanol. We used LC-MS to measure 16 different bile acids in the homogenized intestine and liver samples using mixes produced by Cambridge Isotope Laboratories, and they contained authentic, unlabeled, and deuterated internal standard reference compounds. So our calibrators covered a linear dynamic range of 2.44 to 2,500 nanomoles per liter, and online samples contained a consistent concentration of 225 nanomolar for all deuterated IS compounds. And calibration curves were generated using weighted linear regression models. And I'll mention, just like Andrew said, the method QCs demonstrated stable performance metrics, which provided confidence that the tissue sample data that we collected was efficiently done and correct. So we used, you can see reverse phase chromatography coupled with a scheduled multiple breach monitoring based scan mode on a SIX 6500 Q-trap mass spectrometer operated in negative ion mode was capable of resolving and quantifying isobaric bile acids. And these included beta mucolic acid and cholic acid. So each analyte, which you can see in the chemical formula on the right, has similar fragmentation, and thus we needed chromatographic resolution to differentiate individual analytes. And so isobaric cholic acid and beta mucolic acid, which you can see in the chromatogram, are clearly separately detected. And we also found the same thing for GUDCA, GCDCA, and CDCA, which are all capable of being separately detected by this method. So looking at our data, we observed that there were significantly decreased levels of a number of bile acids in our myo5B knockout mice compared to littermate controls. And these included CA, beta MCA, GCA, TUDCA, and TDCA. And we also saw TCA, DCA, and TLCA, as well as TC-DCA, were all significantly decreased in our myofibrine knockout mice compared to littermate control mice. And interestingly, we didn't see this for all bile acids. There were some that were similar between control and myofibrine knockout mice. And we were unable to detect UDCA, CDCA, and GCDCA in our control and myofibrine knockout mice. And we think this is likely due to the early developmental stage of these mice. These mice are neonatal mice, so they're not fully mature. Mice don't usually mature until 21 days after birth. So these mice are quite young in comparison. So we think older mice would probably have these being able to detected by this method. So overall, we saw a significant decrease in a number of bile acid levels in the liver. And so we next wanted to see what is happening in the ileum, because bile acids that enter the intestine through the duodenum then go down to the ileum, where they are efficiently usually recycled. So interestingly, we saw that we had the reverse in our myofibrine knockout mice, where we actually had significant increases in a number of bile acids in the ileum of our myofibrine knockout mice. And we think that this might be because of the enterohepatic circulation, which usually efficiently recycles. About 95% of the bile acids are taken up by the intestine, transported into the circulation, and then restored to the liver for recycling so that they can be reused. And this wasn't true of all bile acids. We did see that some of them are similar between control and myofibrine knockout mice. And we are also, again, unable to detect UDCA, CDCA, and GCDCA in the intestine, and we think that's developmental stage related. So we found that there was retention of select primary bile acids in the ileum, and we did have some elevation of secondary bile acid, DCA, in our knockout mice compared to control mice. So we think that this is likely true in the ileum because there are bile acids in the lumen normally, and these are transported into the ileal enterocytes by ASBT, which is the primary bile acid transporter in the intestine. So ASBT uptakes bile acids, they enter the cell, and then they're pumped out of the cell in the basolateral membrane by OST alpha and beta, where they enter the circulation and return to the liver, where hepatocytes take them up and then will resecrete them. And this happens about 4 to 12 times a day. So bile acids are highly recycled by the body so that there's not a large load on the hepatocytes for making new bile acids constantly. And ASBT, in the presence of high levels of bile acids, can actually signal through FXR and secrete FGF19 in humans and FGF15 in mice to reduce the level of bile acids that are being made. So that 5% that would be contributed by hepatocytes with the 95% recycling. So we look at our RNA sequencing, and interestingly, we saw increases in ASBT and OST alpha and beta, and increases in FGF15, at least trends within the FGF15 and ASBT. And this suggests that there's changes in the FXR axis that might be impacting have a negative feedback loop on bile acid synthesis. And so since we saw increases in ASBT, this sometimes indicates that there's actually a compensatory mechanism when a protein is mislocalized. So we stained for ASBT, which is in red, and it normally localizes to the apical brush border, which is seen in green. So this is the apical membrane, where bile acids in the lumen would be transported into enterocytes and then exit out the basolateral membrane into the blood. And so ASBT highly co-localizes with gamma-actin on the apical membrane in our control mice. But in our myofibrine knockout mice, we actually see a lot of red intracellularly, and we see very little yellow on the apical membrane, suggesting that we don't have proper localization of ASBT to efficiently recycle the bile acids from the lumen into the tissue and then into the circulation for routing to the liver for recycling. So our data really suggests that loss of myofibrine in an animal model really alters bile acid synthesis genes, as well as, very importantly, alters functional bile acid synthesis that we are identified by LCMS. And we think these results in changes in bile acid pool composition, which really impact both the intestine and the liver. And we think that this is a really great method for looking at, especially in animal models, at bile acid composition, which can be hard to measure, and to look at specifically individual bile acids, which can be hydrophobic or hydrophilic, and some can be more cytotoxic than others. So I'd like to end by thanking the amazing collaborators, Andrew Percy and Thomas Horvath did a large amount, most of the mass spec, all of the mass spec work, I should say, and then my lab, which did the RNA sequencing and immunostaining. So I'm happy to take any questions.
Thank you, Amy, for that excellent overview and rationale into the BA results of the mouse models. I'm very cognizant of the time, and with that, we don't have any time for questions. So we will press forward. If you can stop sharing your screen.